All right, today's lab on the testis slides. We're going to look at a lot of slides here, a whole series of them. There's going to be a lot of redundancy in the terminology, so be ready for that. I'm taking the images today off my computer because it doesn't give that black bar problem that we have with the television set. So here we go. First thing to think about, on the very outside of the testis, there's a thick layer of connective tissue. You see it here across the top of the screen. It's mostly pink and has in there a lot of fibroblast cells and mostly collagen. It's kind of a wavy collagen, kind of not a very regular wave pattern to it, not like what you'd see in a dense regular connective tissue or a nerve fiber, but anyway, it's kind of a mixed up wavy collagen covering. It's a dense connective tissue. It's called the tunica albuginea because there's very little blood supply to it, and it's very white in its overall appearance. Inside of that we have all of the seminiferous tubules in there. You can see these circular structures here, rounded structures. Sometimes we don't cut straight through them, we catch them at an angle, so we see elongated or oblong shapes of seminiferous tubules. They will be surrounded by peritubular cells, and between the seminiferous tubules we're going to see bits of connective tissue, blood vessels, and the Leydig cells, the interstitial Leydig cells that produce testosterone. Another image here of that dense connective tissue of the tunica albuginea. There on the left, a seminiferous tubule on the right, seminiferous tubule over here on the left. So lots of fibroblast nuclei in purple, and then the collagen is that kind of reddish brown color. Our next tunica albuginea slide is at a lower magnification, so we can see the entire thickness of the tunica albuginea in this case. So down in here we have the seminiferous tubules, and then all of this is tunica albuginea. On the very outside there is a mesothelium, simple squamous cells. A part of your peritoneal cavity extends down to the scrotal sac and wraps partly around the testis there, forming a little extension of your body cavity there. So that's still part of your body cavity lining, so you have a mesothelium on the outside. It doesn't completely encircle the testis, so on the opposite wall from this you probably would not see any mesothelium there. Magnify that mesothelium a little bit here with my computer, so you see it right on that edge, right along the outside there. The next slide here is to show the Leydig cells that are in between the seminiferous tubules. So here's a seminiferous tubule wall here. These nuclei in here are peritubular nuclei, so each of these is a seminiferous tubule. We have 4 seminiferous tubules, and this is in between, and that's kind of in between over there. You can see a little bit of the collagen fiber, some other cells over here. But anyway, right here we have Leydig cells, these nice round nuclei here, and one right here, another one right here, a couple more here. So these are the interstitial cells that produce testosterone. Let's look at another example. Slightly different stain on this slide, but here we have a little clump of Leydig cells. Looks like 5 or 6 or more of those Leydig cells there. Now outside of the Leydig cells we have a couple of fibroblast nuclei here. Can't see the collagen very well in that slide. And of course these are peritubular cells around the outside of the seminiferous tubules, and we're going to be looking eventually at all of the cell types inside of the seminiferous tubule. Another look at Leydig cells here. So here's seminiferous tubule, there's an artery down here, and this is a clump of Leydig cells. There's some more over here. Those nice round nuclei. The cell shape, it's not exactly round typically, but anyhow, there's your interstitial cells of Leydig, sometimes simply called interstitial cells, sometimes called cells of Leydig. This slide is intended to just show the peritubular nuclei that surround the seminiferous tubule. So we've got quite a few of them in there. They look very much like smooth muscle cells. The nucleus of the peritubular cell is about the same shape as a smooth muscle cell nucleus. So this is interstitial tissue over here in between. So there's little bits of collagen, some fibroblast cells. Here's a interstitial cell of Leydig here. Looks like a little artery right over here on this side. More of the same. Here we have these peritubular cell nuclei right in a row. There are more down here on this wall along that side of the outside of the seminiferous tubule. Interstitial tissue here, a couple of Leydig cells in between. Now we're going to go inside of the seminiferous tubule and look at Sertoli cells. Sertoli cells are different from all the other cells inside of the seminiferous tubule. These have an odd shape. You can see one right here. It's kind of an elongated. It's not a very organized kind of shape, but they're very tall cells. There's 1, 2, 3, 4. There's one kind of out of focus right here. So anyway, these are Sertoli cells. Remember, they have a kind of an assisting role in the testis. They are not part of that spermatogenesis process. They sequester testosterone. They form that blood-testis barrier. They contribute nutrients to the developing spermatocytes, and they recycle the excess cytoplasm that's sloughed off of sperm. When we look for them, they're pretty consistent in having a nice oval nucleus with a distinct nucleolus in there, and their cells just kind of look like ragged sheets pulled out away from there. It's almost like peeling out saran wrap or something like that in the shape. But you can often see the margins or separations between the individual cells. Our next Sertoli cell example. Here's several in a row. They don't always occur in a nice sequence like this, but all of these are Sertoli cells. There's a couple of spermatogonia nuclei back here that we can see behind them. Another view, let's magnify that a little bit. This one right here is real good. You can see that it extends from here all the way out to here. There's one side along there, and then there's another side right along here. So here's the next Sertoli cell. This is one of the spermatogonia. This is a spermatogonium, so they're down here next to the wall. The outer perimeter is where you find spermatogonium. Those we'll talk about here in a minute, but anyway, these are Sertoli cells right through here. Another couple down here in the picture. The next example of Sertoli cells, we have them right in here. Nice nucleus here and here. You can see the boundaries to that. Here's another nucleus out here, kind of out of focus. But anyway, a few Sertoli cells in a row, and these are out at the base, out by the peritubular cells that we have out here. We have spermatogonia. Our next Sertoli cell example here, we've got a couple over here on this side and a few down here at this base. This isn't quite as good. I think we'll just go ahead and move on to the next one. In this example, I'm not sure exactly why, but we don't have any spermatogonia along here. So these are all Sertoli cells in a row. They have that similar nucleus with a nucleolus in the center. And you can see on some of them the distinct border between the cells, dividing them up. So it's kind of like a columnar epithelium, but very, very ragged looking. So those are Sertoli cells. Next, we're going to look at seminiferous tubules in general, to look at how there's kind of a progression of cells from the outside toward the middle here. So the ones on the very outside are always spermatogonia, these nice round nuclei here. And as they divide, those cells that divide will form some primary spermatocytes and some secondary spermatocytes that are kind of larger in size. So these are probably secondary spermatocytes. I don't see a primary here. That's obvious anyhow. And eventually it'll form spermatids, these smaller cells right here. These are spermatids right here. And then we see the heads of some sperm out here toward the middle. So as they divide, they move more and more toward the center of the seminiferous tubule. Another example, you have the same thing here. You've got on the very outside, you have these spermatogonia out here. They divide and the cells move in toward the middle. Now these are probably a primary spermatocyte right here. In that example, that one there. They could be secondary, however. These are obviously secondary spermatocytes right along here. They have a smaller size. And not a lot of spermatids evident here, but several of the developing sperm in here. At this point, it's a good thing to know that the stage of spermatogenesis as you move from the outside to the inside is pretty consistent in any particular location. So if you're looking at one spot on a seminiferous tubule, all of the cells will be in about the same stages of development, or in the same sequence. So in this one, we don't have many spermatids. In another slice, we would be almost all spermatids in the middle of the structure. So let's go on to the next one. Oh, this one doesn't have much going on in here. There's kind of a clump of sloughed off cells in the middle of the tubule. Around the edges, we have the spermatogonia right out here. Lots of Sertoli cells. These are all Sertoli cell nuclei along this side. These are probably secondary spermatocytes. These looks like about 6 right here, a couple out there in the middle. There are a few spermatids in here and a few developing sperm. There's a set of 4 right there, developing sperm. And we usually, well, it's very common to see sets of 4 developing sperm or spermatids, because the 4 result from the meiotic separation of one primary spermatocyte. Another seminiferous tubule. Again, we have the nice peritubular cells out here on the outside. We have interstitial tissues here. There's some Leydig cells going on there. We have spermatogonia out and around the margins. We have primary and secondary spermatocytes. Again, it's sometimes difficult to tell what stage they're in, whether they are primary or secondary if they're not extremely large. The largest ones we'll assume are going to be primary spermatocytes, but these seem to be secondary. These are spermatids out here. They haven't entered in that stage of transformation to form sperm. There are a couple of developing sperm, maturing sperm right up in here. In this one, we have picked up an artery right here. We've got a small artery. We have some interstitial tissue out here. I don't see, yeah, it looks like a Leydig cell way up there in that corner. We're going to magnify this a little bit. See this seminiferous tubule. The small dots here are all sperm. This is developing sperm. You can see the beginnings of its tail coming out there.
These are developing sperm, maturing sperm up here. So we have spermatogonia out on the outside. Here's a Sertoli cell and another one there kind of strung out. There's a nucleus of a Sertoli cell there and there and there. So anyway, we have some of the stages. Here's a very small spermatid right here, right here, and right here. Notice how much smaller the nucleus is in a spermatid compared to a secondary spermatocyte. It looks like this is a late stage of separation of a primary spermatocyte into two secondary spermatocytes. It's a little bit odd that the nuclei are so organized at this stage before splitting the two. It may just be an aberrant cell, but you can tell that the cytoplasm, the whole thing, is a lot larger than a single secondary spermatocyte there. So they have to get large and then they split. This one illustrates stages of spermatogenesis. Again, we have a nice seminiferous tubule we're going to concentrate on here, peritubular cells on the margin, spermatogonia out here making like a dots along the outside, big dots. And we have a nice big cell right here. That's almost certainly a primary spermatocyte. We're going to assume that's a primary spermatocyte. These down here would be secondary spermatocytes. And then we also have some spermatids in here. Let me magnify that a little more. Might as well. Here we go. So our spermatogonia on the outside, our primary spermatocyte here, secondary spermatocytes down here. These are all spermatids, little clusters of them in here. And not many of them are starting that transformation to form mature sperm. There's a small developing sperm head there, and looks like one's beginning to form there. Another view of a primary spermatocyte. Because of its large size right here, we assume that to be a primary spermatocyte compared to these cells up here, which are probably all secondary spermatocytes, and then these are spermatid cells right in through here. Again, I should magnify that before I talk about it, but anyway, that's the, whoa. Another look at spermatogenesis. This looks like a secondary spermatocyte has divided, the nuclei are divided, and the cytoplasm is still together in that one. Another primary spermatocyte, very large size, really stands out. These would be the spermatogonia out here around the margin. There's a Sertoli cell nucleus here. Some secondary spermatocytes in there, but lots of spermatids already formed in this view. Here we have an abundance of secondary spermatocytes. All these along here, there's a single primary spermatocyte right there. Let's magnify all this a little bit. So we know those that are going to be out here against the peritubular cells out along the outside wall of the seminiferous tubule. These will be spermatogonia. Here's a couple of Sertoli cell nuclei. These are secondary spermatocytes. That's a primary spermatocyte. And over here we have the smaller cells or spermatids. Some developing sperm heads right through here. Start to see some tails in here as well. In this image, there's an abundance of spermatids. All these in here are spermatids. These are all about the same stage. So out on the outside, we have the spermatogonia that divide to make primary spermatocytes, the first step to moving something towards spermatogenesis. The primary spermatocyte, remember, is the one that undergoes the first meiotic division to form secondary spermatocytes. Here's some secondary spermatocytes over on this side.
lots of spermatids. All of these will eventually transform into maturing sperm. And that's the next thing to look at, maturing sperm. This is a pretty good illustration. We see the pointed end to the sperm head here, and we see this kind of blurry mass that kind of follows behind it. And that's true for all of these right in here. These have not completely lost their cytoplasm yet. They still maintain some of their cytoplasm that will be sloughed off. It'll be lost. That's called the residual body. So they're losing their residual bodies. And that residual body material, a lot of this stuff you see out here is residual body material. It will be gathered up by Sertoli cells for recycling and using as nutrients to make more. This is primary spermatocyte down here. Another view of maturing sperm. Let's see if we can get some of this in here. Here you see the head and the residual body here. There's some little tails, little wispy tails going through there, but here's another residual body mass up here. So those are maturing sperm. You still have some of their cytoplasm hanging on. Spermatozoa, maturing spermatozoa. Here we've lost our residual body. It's all gone. We have the head here and we have a tail that can't really tell which direction it goes. There's a couple of tails overlapping there, but anyway, there's a sperm. They are not fully motile while in the testis. They are getting there, but they're not. They develop their full motility after they've left the testis. Here's another. Seems to still have a little material on there, which is odd, but anyway, that's a sperm there. Let's look at another example. Here's a whole cluster of them up here. All of these here are maturing sperm, or mature sperm. We call them spermatozoa. We can see their long tails extending back behind them here. So these are mature sperm. But they don't have their full motility yet. Here's another image. There's just a few right in here in this center. So here's the head and here's the tail. There's another head and tail. It's kind of hard to see the tail on that one. The other cells around here, these large ones would be secondary spermatocytes. I don't see, there's a spermatid down in this corner. A few more spermatozoa here. Good example right here. They see the head of the sperm and then the tail behind it. There's some tails here, but which sperm head that goes with is a little hard to see. Spermatogonia over here on the side. These would be peritubular cells way over on this side. Spermatids, these smaller cells. See how these are smaller than the secondary spermatocytes nearby. Good example of spermatid right here as well. Now we're going to move on to near the end of the seminiferous tubules, where we see that we no longer have sperm development going on around the margin. No spermatogonia, no Sertoli cells, no other cells attached to the wall. This material is just kind of passing through this tube. This section we call the straight tubule. The straight tubule. So that's the very last part of the seminiferous tubule before you go into the rete testis. So these are very low cuboidal, kind of granular cuboidal cells. These are even lower up here in this one. See how they're almost squamous cells. We'll still consider them low cuboidal cells, what we'll call them, but they're flattened quite a bit. Another view of a straight tubule, same kind of thing, very low cuboidal cells around the margins, no ring of Sertoli cells or spermatogonia. Here we have several different cells, spermatids and secondary spermatocytes, it looks like, that are being pushed into that part. And our next straight tubule example, again, these low cuboidal cells around the margin and materials flowing through, passing through that straight tubule toward the rete testis. Now, if we were in the rete testis, we would have a similar kind of epithelium, but we would not have the tubes surrounded by seminiferous tubules. So in the next example we go to, well, let me go back one. We have one more example here to look at of a straight tubule where we see the transition between seminiferous tubule and a straight tubule here. So this is a good example to look at. You go back a little bit in magnification. Over here we still have Sertoli cells and spermatogonia and all that going on the outside of the tube. And as we progress this way, we see this change over to those cuboidal epithelial cells on the margin. So this is the beginning of the straight tubule, or the tubuli recti, is another name for them, straight tubules of the testis. So that's that transition from seminiferous tubule to straight tubule. Now we'll look at rete testis. Now we're surrounded by collagen, so we're in a dense connective tissue region, and this is same kind of epithelium essentially, that low cuboidal epithelium, very small cells that surround the tubes of the rete testis. Another view of rete testis, a little lower magnification, so you can see it forms a network of tubes through those connective tissues in here. So you can see the branching and coming together of tubes, this network, this maze of passages between the straight tubules and the ductus deferens, not the ductus deferens, the efferent ductules, that's what I need to say. Between the straight tubules and the efferent ductules or tubules. This is all rete testis, all these passages, and they all have that same kind of low cuboidal epithelium. So that's the testis. They've seen all the tissues of the testis.